Hello viewers, this is Dr. P. Mary Anupama. After seeing nitrogen cycle and then nitrogen fixation, let us talk about assimilation of nitrogen. First, let us see the intake of nitrogen from the soil by the plants. We know that plants they cannot fix the nitrogen while they absorb the fixed nitrogen from the soil. The bacteria, whatever they fix, or the algae, whatever they fix, this is taken up by the plants from the soil. So you have four kinds of compounds which are available in the soil. So we have nitrates, nitrites, ammonium salts, as well as some organic compounds. Plants they can utilize nitrates as a nitrogen source. Some plants they rarely take up nitrites, while some plants like rice they can take up even ammonia. So ammonium ions they can be absorbed very easily through the roots. And you have ammonium nitrates. These are considered the best source of nitrogen, ammonium nitrates. People also add urea, right? Urea is the organic compound and this can also be absorbed by the plants very easily. Reduction of nitrate in plant tissues. The nitrates from the soil, these are converted into ammonia source and then to amino acids. The nitrates reduction into ammonia it takes place in the roots as well as in the leaves, and this reduction process it requires gain of electrons. So here you can see that the three atoms of oxygen and one atom of nitrogen they will require around 6 electrons or around 8 protons, that is 8 hydrogens, to form 3 molecules of water and 1 molecule of ammonia. So if you look at the overall reduction process from nitrate to ammonia, it is taking place in 4 steps and each step it's consuming 2 electrons. So plus 5 to plus 3 state, then to plus 1 state, and then again minus 1 and minus 3. So this is how it is being converted to ammonia. These are the steps. So first nitrate is converted into nitrite, then nitrite to hyponitrite, hyponitrite to hydroxylamine, hydroxylamine to ammonia. And the reactions involved they are as follows. The nitrate is converted to nitrite. NADPH is a source for protons and you have nitrate reductase. Then the nitrite is reduced to ammonium hydroxide. You have nitrate reductase again doing the same thing, then 2 NADPH. Then finally you have ammonia in the last step formed, and this is also catalyzed by nitrate reductase, and finally one water molecule is formed. So NADPH, NADH, these are the sources for protons. Nitrate reductase it catalyzes the reaction of this reduction and it requires some metalloflavoproteins like NADPH2 or NADH2, and it also requires some electron acceptors like flavonidine dinucleotide and some prosthetic groups, and even molybdenum that is required as a cofactor. So the electrons they are transferred from NADPH or NADH2 to FAD. FAD is reduced, and then even this molybdenum, reduced molybdenum, it gets oxidized and protons are released during this process, and the nitrate is converted to nitrite. So this is how sequence of reactions will be taking place, and finally ammonia is produced. So assimilation of nitrogen in the synthesis of amino acids. Amines, amino acids, these are the initial products of this nitrogen assimilation. We have this alpha keto acids, which are the ones which receive this amino group, and then the keto acid is converted to glutamic acid. So there are two ways for this other amino acid synthesis that take place. You have reduction amination and transamination that takes place. Okay, reduction amination and transamination, these are the two reactions which take place by which the nitrogen is assimilated into all the amino acids. The first one is reduction amination. So ammonia, which is formed by the reduction of nitrate, it combines with the alpha ketoglutarate acid, which comes from the Krebs cycle, and then it forms the glutamic acid finally. So this reaction, it is reversible reaction, and it occurs in two steps. Okay, the first step is spontaneous, while the second one, it requires the enzyme glutamic dehydrogenase and then NADH2. Okay, so this is first alpha ketoglutarate acid, which is spontaneously converted to alpha aminoglutaric acid as it takes up ammonia. Now the next step of conversion of alpha aminoglutaric acid to glutamic acid, it requires the enzyme glutamic dehydrogenase and NADH2, and finally our glutamic acid is formed. This is called as the reduction amination because protons are incorporated. So it is very rare that other than glutamic acid, other amino acids, they undergo this reduction amination. You have this oxaloacetic acid, which takes up ammonia to form aspartic acid. Fumaric acid, it gets converted to aspartic acid. Pyruvic acid, it gets converted to alanine by reduction amination. The next one is the transamination reaction by transaminases. So transaminases, the name itself indicates that they are involved in the transfer of amino group. So amino group from one amino acid to the other amino acid. So it is transferred to another keto acid basically. So in most of these reactions, what you see is that the glutamic acid, it is the donor for the amino group. Say for example here glutamic acid, it donates the amino group to oxaloacetic acid, and we have seen that oxaloacetic acid gets converted to aspartic acid, while the glutamic acid, it forms alpha ketoglutarate acid, which is ready for another reduction amination reaction. We also have glutamic acid, which transfers amino group to pyruvic acid, alanine is produced, and alpha ketoglutarate acid is produced. So these transaminases, these are found in the mitochondria, and they require pyridoxal phosphate. Pyridoxal phosphate is involved in the transfer of amino group. We also have synthesis of amides, which is taking place, amides, that is asparagine and glutamine. So these amides, these are important for the storage and transport of nitrogen within the plants. So the hydroxyl group of the carboxyl group, it is replaced with amino group. So here you can see alpha ketoglutarate acid, wherein it takes up ammonia in the presence of ATP. In the presence of this enzyme glutamine synthetase, it is converted into glutamine, and this glutamine synthetase, it requires magnesium as cofactor as well as ATP. ATP is required. So in this you see alpha ketoglutarate acid, it is converted into glutamic acid, and then the glutamic acid, it is converted into glutamine by glutamine synthetase. Coming to glutamine synthetase, its enzyme commission number is 6312, and it plays important role in the nitrogen metabolism. It catalyzes the condensation of glutamic acid and ammonia to form glutamine. And you see that ammonia, it comes from the nitrate reduction and the amino acid degradation, or even from photorespiration. So the concentration of this ammonium ions, it's mainly responsible for this glutamine synthesis and even glutamine hydrolysis, the concentration of ammonium ions. So the glutamine synthetase, it catalyzes this ATP dependent condensation of glutamate to glutamine to yield glutamine. So ATP hydrolysis, it drives the first step, wherein the ATP, it phosphorylates the glutamate to form this acyl phosphate intermediate, and then you have this ammonia which steps in here, and here the phosphate is replaced by ammonia, inorganic phosphate is released, and here ammonia gets attached, and finally you get glutamine synthesis taking place. Let us see the classes of glutamine synthetase. So there are three classes of it. Class 1, class 2 and class 3. Class 1 you find in the prokaryotes, and these are oligomers which consist of around 12 identical subunits. And the activity of this GL1 type 1 enzyme, it is controlled by adenylation of the tyrosine residue of this particular enzyme. So adenylated enzyme, it is considered to be inactive. If it is not adenylated, that is the tyrosine residue, then it is an active form. Coming to class 2 enzymes, we find them in the eukaryotes and even in some bacteria that belong to this Rhizobiaceae, Franchiaceae and Streptomycetaceae families. These are decamers of identical subunits. While these have 12 subunits, dodeca, these are decamers. And plants, these have 2 or more isozymes of GS2. One isozyme, it is translocated in the chloroplast, while the other one is found in the cytosolic area. The cytosolic glutamine synthetase gene translation, it is regulated by 5' untranslated region, the UTR region, while its 3' UTR, it plays a role in transcript turnover. Coming to class 3, it has been found in bacteroids and Butyri vibrio species, and it is a double ring dodecamer. It is a dodecamer, but double ring dodecamer of identical chains again. It is very much larger, above 700 amino acids. Here GS1, it is a dodecamer, but it has 450 to 470 amino acids, 450 to 470. This one 350 to 420, while this one it is very huge, 700 amino acids. Now let us see the regulation of glutamine synthetase. Glutamine synthetase, it is subjected to this reversible covalent modification. That means it is covalently bond, as well as again that molecule which is attached is removed, that is adenylation and deadenylation. So it consists of 12 subunits, each having one tyrosine residue, tyrosine 397. Now here the glutamine synthetase, it is undergoing adenylation as well as deadenylation by the enzyme adenylyl transferase. That is, same enzyme is doing both the things, so it is bifunctional regulatory enzyme. Adenylation, it's a post-translation modification wherein you have AMP attached to the protein side chains. Each adenylation, it requires one ATP and complete inhibition of one glutamine synthetase. It requires 12 ATP because each subunit has one tyrosine. So 12 subunits have 12 tyrosine residues, and each residue is to be adenylated by AMP. So obviously it requires 12 ATP. Deadenylation by it involves phosphorylatic removal of the tyrosine linked adenylyl residue as ADP. If this is taking place, what's happening is there is inorganic phosphorylatic removal of tyrosine. It requires the conversion of AMP into ATP, which is taking place. This AT activity, it's influenced by another regulatory protein that is associated with the P Roman 2, we call it P2, which is a 44 kD trimer. It's a trimer. So this activity, adenylyl transferase deadenylation activity, is regulated by this P2, and this P2, it undergoes post-translational modification again by uridyl transferase. So here you have uridyl transferase, which is influencing P2. It is involved in the post-translation modification of P2. So you see that P2 also exists in two forms. It also exists in two forms. So the state of this P2, it dictates the activity of this adenyl transferase. So P2 is not uridylated, then it will be existing in the form of P2A, the non-uridylated P2, it's called as P2A form. Okay, the other one is uridylated form is P2B. D form. Okay. ATP2A complex, it will deactivate the GS, the glutamine synthetase, by adenylation. And if P2 is deuridylated, then it will take up the form of P2D. If P2 is uridylated, then it will take up the form of P2D. And this ATP2D complex, this will activate the glutamine synthetase by deadenylation again, by deadenylation. So this P2D, it is deadenylating the glutamine transferase. Now it is, glutamine synthetase. Now this is our active glutamine synthetase. So this ATP2A and ATP2D complexes, these are allosterically regulated by mainly our alpha ketoglutarate and glutamine based upon their levels. So glutamine GLN, it will activate ATP2A activity. That means this will not contribute to the formation of glutamine because already glutamine is there. So it will inhibit the formation of P2D, thereby inhibit the work of glutamine synthetase. No more it is required. Okay. So the effect of alpha ketoglutarate on the complex, it is the opposite. That means if alpha ketoglutarate is there, it is ready to accept more of ammonia. Okay. And it is ready to accept ammonia, it is ready to get converted into glutamic acid and then into glutamine. So therefore you have this P2D formation. So ATP2D formation will be taking place. Because of this, the glutamine synthetase, it is converted into active form by deadenylation, and then finally glutamine synthesis is taking place. Coming to the inhibitors of this enzyme, inhibitors are a result of this glutamine metabolism. So you have this tryptophan, histidine, carbamyl phosphate, glucosamine 6-phosphate, cytidine triphosphate, adenosine monophosphate, all these are inhibitors. Others inhibitors or regulators include glycine and alanine. So here you have glycine, alanine and serine, glycine, alanine and serine. These bind to the glutamate substrate site. So you have GDP, AMP, ADP. They bind to the ATP site. So here you can see the activity of P2 when there is high nitrogen and when there is low nitrogen. If there is high nitrogen, obviously there is a need to convert this P2 UMP, the uridylated one, to P2, and then the glutamine synthetase, it becomes active. Okay, it is involved there, glutamine synthetase, and then you find glutamine synthesis taking place. Now if nitrogen is very low, there is no need for glutamine synthetase to be there, to be in active form. Then it is retained in inactive form. It is adenylated and it is retained in inactive form. So high nitrogen, it leads to this. Low nitrogen, it leads to this. For any of these things, you require ATP. So with this, we complete the nitrogen metabolism. In total 3 episodes are there. Do watch them. Thank you.